hottest drugs and specializes in GI malignancies. He's a wonderful resource and an expert in the newest and hottest drugs. Dr. Cleary.
Thanks. Well, I'm really excited to be here. I just wanted to take a moment. Peter's talk actually was really good. I actually learned some things listening to him. But in addition to being an outstanding clinician, Peter runs the whole gastroesophageal effort here. He's been a wonderful mentor to me and really has brought along, in addition to me, a lot of junior faculty, which has made a big, big difference. So I'm very grateful for Peter. What I'm going to be talking about today is navigating clinical trials. And I really wanted, since we're a small group, I wanted to invite anyone who has a question while I'm talking just to raise your hand, and we could sort of just talk about anything that comes up. Really what clinical trials are, as Peter was just saying, are a way to take ideas from the laboratory. So for example, Adam Bass runs a great laboratory developing new therapies for gastroesophageal cancer. So for taking ideas that scientists like Dr. Bass generate and then trying to bring them to the clinic, trying to figure out a way to see if these ideas that seemed promising in the laboratory actually help our patients. And the structure of clinical trials, you'll hear a lot of talk about different phases of trials, and I think it's important just to sort of talk about what these words mean. So again, when these ideas start, they start in laboratories. They start in laboratories at institutions like this, at academic centers like Dana-Farber, but also in big pharmaceutical companies like Novartis and Genentech. And so what these scientists are doing is they're trying to figure out chemicals that seem to be promising in fighting cancer. And a lot of time goes into figuring out which chemicals are promising in fighting cancers. Once these scientists have felt like, wow, we have a promising compound, then we bring it into the clinic. And their first step in the clinic is something called a phase 1 trial. And what a phase 1 trial is, as you can imagine, is because we're trying it for the first time in human beings, we really want to see, is this drug safe? Is this drug causing any harm? Because as a physician, the last thing we want to do is cause any harm. So we want to think about, A, what the side effects are, B, what the highest safe dose is. But also in these phase 1 trials, we also want to get a sense of, does this drug actually work? Because at the end of the day, that's what we care about. So we're starting to get a sense of, A, is the drug safe? What are the side effects? What's the right dose? But B, we're starting to get a little idea about, does this drug have any efficacy? But more formally, where we test efficacy is in phase 2 and phase 3. Phase 2 is a smaller trial than phase 3, where we just start to say, okay, we're only going to give this drug to people with esophageal cancer. And compared to other trials out there, how does it work? Does it seem promising? But phase 3 is actually the most statistically rigorous trial. So phase 3 are these very large efforts where we can stay with a lot of statistical rigor that this therapy is better than any existing therapy out there. So when I think about drug development, I think about step 1 in the laboratory, finding promising compounds. Step 2, doing a phase 1 trial, seeing what drugs, seeing if a drug has any serious side effects, seeing what the right dose is, and seeing if it's safe. And then in phase 2 and phase 3, actually figuring out, does this drug actually work? Does this drug make people live longer? And if after phase 1, 2, and 3, we figure out the drug is reasonably safe and improves survival, the FDA approves it. And in terms of this process, I'd like to point out that this process works. Yes, there are a lot of failures, but actually there are some success stories. So in Peter's talk, he talked about ramucirumab, and in a trial actually that was led by Charlie Fuchs, who was working here at the time, he found that ramucirumab prolonged survival in gastroesophageal cancer patients. So this process does work. But one of the dangers of this process is we are using human beings as research subjects. So a real important thing when we're doing these clinical trials is having ethical oversight just to make sure that we're not putting people at higher risk than necessary. And one of the ways they figured out that this was necessary was there some terrible stories. Like some of you might have heard about the Tuskegee syphilis study. And what happened here is before there was a cure for syphilis, they wanted to see what happened with people with syphilis, what sort of medical problems popped up. And what they found was that they were studying this, but then a cure came out for syphilis. And the big ethical disaster that happened on this was basically when the cure came out for syphilis, they did give it to people on the trials. So once they saw this disaster, they realized that there had to be really strict ethical oversight. Unfortunately, we now have that. So thanks to something called the Belmont Report, every institution, every clinical trial is subject to something called an IRB. And when I think about an IRB, I basically think of it as the ethical board. So basically, it's a group of scientists, clinicians, patient advocates who get together to say, is this study ethical? And so for any clinical trial here at the Farber, there's a few steps that the trial has to go through before the study gets approved. Step one is that the scientific community has to say, is this study even worth doing? Is it asking an important question? Is it going to help our patients? Step two, though, is the IRB, the ethics board, says, is this fair to our patients? Are we putting our patients at too much risk? Are we doing everything possible to make sure that the patient's safety is being maximized? And then once we get through all those committees, then the trial starts, and then we can start putting patients on. In terms of the approaches we're doing, I would say in general there's two big approaches. One approach is a personalized medicine approach. And what I think about this is, when you think about a cancer like gastroesophageal cancer that happens to so many different people, you start to realize that all the gastroesophageal cancers aren't equal, and that different people with gastroesophageal cancer have different markers. And so a big push has been to try to identify markers within a cancer like gastroesophageal cancer so you can get the right patient to the right therapy. So for example, Peter mentioned before a trial called the TOGA trial. And on this TOGA trial, we only took a fraction of people with gastroesophageal cancer, only about 15%, because we knew the other 85% it didn't have a chance of working. But in that 15% who had a certain marker called HER2, we said, oh, maybe this antibody we have directed against HER2 could help them. And in fact, it did. And that idea of trying to find a marker that could be targeted with a therapy is really driving a lot of our work. And to do that, we have to know what the markers are. So in order to get on that TOGA trial, you had to do a test, a molecular test, to find out that you had this protein HER2. And we really strongly believe in this approach. And what you'll see from the rest of this talk is a lot of our trials are looking for these markers. We want to say, okay, we know the patient has gastroesophageal cancer, but what other markers do they have? Another marker Peter mentioned in his talk was MSI high. And so basically we want to see as many of these markers as possible so that we can try to find weaknesses in the cancer that are suggested by that marker to get the person on the right therapy. So to do this at the Farber, we're very lucky. We have a philanthropically supported program that allows us to take patients' tumors that have already been removed from their body and do sequencing on them. And really what that means is we just want to know what mutations are happening in that patient's cancer. And occasionally when we see these mutations, we'll say, oh, we have an experimental drug that patient might benefit from. Or that this patient has a lot of HER2, so we should put them on the TOGA trial approach. So that's why this molecular sequencing has helped us so much. And to kind of give you an idea of the power of this story, it actually comes from a story from lung cancer. So right around the time when I started in oncology, 2006, there was a drug called crizotinib, and people were very interested in this drug because it hit a marker, a protein called MET. And everyone really felt like MET, which we still think is a very powerful driver of cancers, that if we had a drug against MET, it would really slow down the cancer. Well, it turned out, unfortunately, the trial didn't go so well. So initially, 2006, they put a lot of people on this drug, and no one benefited. It really was, unfortunately, a bust. But then, unexpectedly, in Japan, they found this discovery that a group of lung cancer patients have a marker that we didn't even know about at the time, called EML4 ALK translocation, but basically had a marker that they found we just didn't know about. And all of a sudden, these investigators doing the trial said, well, you know, we didn't think the ALK part of this drug was that exciting, but now that we heard about this discovery, maybe we should only put the patients with this new marker, the EML4 ALK translocation, on this drug. And really what really, to me, makes this such a powerful story is this drug went from something that didn't help anybody to once they started putting the right patients on the trial, the patients with this translocation, it really just ramped up very, very rapidly. So what happened is when they put the lung cancer patients on with this marker, all of a sudden they started seeing lots of responses. The response rate was over 50%. So it went from 0% to over 50%. And really, and this is why this is a success story in drug development. The discovery was made in 2007. They got the first patient on in 2008, and the drug got FDA approved in 2011. That's the way we'd like to see drug development go, that a discovery happens, we have a therapy for it, and it gets FDA approved rapidly. I wish I could say that was always the case, but this really is what we're working towards. And to show you really what has happened, so this is just an example. So this X-ray here, you can see there, I don't know if you can see my pointer. Here, I'll start to point to it. Right here is where the tumor was, and then really the tumor just shrunk away here. So it's very, very impressive. And in this graph down here, all the bars underneath this line indicate patients who had tumors get smaller. So as you can see, there really was only like 5 patients on the trial. The tumors got larger that go above the line, and most of the patients went below the line, just showing the power of this approach. And the other thing I would just sort of point out is that we've seen examples of these targeted therapies where this drug is approved in lung cancer, but we'll see examples where also it'll work in other cancers like lymphoma. As a matter of fact, I had a patient with gastric cancer that had a lot of copies of the MET gene I gave this drug to, and it worked for 4 months. So we've sometimes seen a drug approved for one cancer, lung cancer, when a patient with gastric cancer has the same abnormalities that the drug hits, it'll work in gastric cancer. And that's something that gets us excited in molecular medicine. The other thing, as Peter talked about, is immunotherapy. And immunotherapy has really taken the oncology world by storm. I like to think about oncologists as oftentimes copying patients of doctors in infectious disease, because in many ways the first chemotherapy was an antibiotic. That was really the proof of principle that you can give a drug that hits helps a disease. And in microbiology, they found that when you're treating tuberculosis, you don't give one drug, you give 4 drugs. So oncologists got that idea. And as Peter was talking about in his talk, we don't usually give one chemotherapy, we give multiple chemotherapies, because we found by giving multiple chemotherapies that kill the cancer in different ways, it's more effective. Again, just learning from our infectious disease colleagues. So the other thing infectious disease doctors have known for a long, long time is the power of the immune system. And I always think about the story of smallpox, how in the 1400s, smallpox was much, much worse than Ebola is even today. There was hundreds of thousands of people dying. It was horrible. And some very clever doctors at the time noticed that milkmaids who worked with cows didn't get smallpox, and they were all fascinated by this. What is this? Well, today we know that milkmaids were getting a very similar virus called cowpox that actually looks just like smallpox. It just doesn't cause the fulminant disease. And they realized, the doctors realized, that these milkmaids getting cowpox were somehow protected from smallpox. And that's what gave them the idea for vaccine. And actually vaccine comes from the word cow in Latin. And so I think probably one of the greatest achievements in medical history, people started vaccinating against smallpox. So this started with a man named Edward Jenner in 1796. But by 1977, through the power of the immune system, through vaccines, smallpox was eradicated from people on the earth. And really that sort of suggested that the immune system can really do powerful things. And as an oncologist, we say, well, wow, if we could use the immune system in the same way, this would be wonderful. And where the immune system, immune therapy has started in cancer, started in about 2012, where scientists started to think about, well, why doesn't the immune system attack cancers? And I should say that a little differently. Scientists have been trying to figure out how to get the immune system to attack cancer for decades, but really it was in 2012 where it started working. And really what the observation was is that ordinarily the immune system should kill off a cancer. So for cancer to prosper in a human being's body, the cancer has to be clever and has to figure out a way to evade the immune system. And so what they'll do is the cancer will express proteins that will protect it from the immune system, that will camouflage it from the immune system. And you might wonder, why would the human body have proteins that tell the immune system to stop? And this also gets back to what Peter was talking about before with autoimmunity, that the immune system has a very challenging job. The immune system has to attack foreign things like bacteria, but can't attack our hearts, because if it attacks our hearts, that would cause us to be very sick. So what our bodies are designed to is things like like your heart, has proteins on them that tell the immune system, don't attack me, whereas a bacteria ideally won't have that, and our immune system will kill the bacteria. So what a successful cancer does is it also co-opts these proteins that tells the immune system to stop attacking. And so what these therapies have done is they've realized what some of these proteins are that tell the immune system to stop attacking, and they make them not function. So really what happens, and what this diagram tries to show, is that ordinarily there's a protein on a cancer that tells the immune system don't attack, and the cancer is allowed to grow. But with these new drugs, and many of them are called PD-1, like Keytruda, pembrolizumab, and nivolumab, that Dr. Enziger just talked about, what these new drugs do is they block the function of these cloaking proteins, and by blocking their function, it allows the immune cells to go in and kill the cancer again. And this is just a diagram that shows us, we call them checkpoint inhibitors. And the reason we call them this is you almost can think of it as ordinarily in the body, this checkpoint is down and the immune system's protected, but by using these checkpoint inhibitors, it opens up the checkpoint and allows the immune cells to go in and kill off the cancer. And again, as Peter pointed out before, the reason we're so excited about immune therapies is it really changed the way we think about cancer. Oftentimes, we like to think about therapies that are successful in improving survival by a few months, which is important, which is very important. But in immune therapy, when it works, because as Peter pointed out, unfortunately, it doesn't work nearly enough. It only works in a small fraction of patients. But when it works, it really can be a game changer, that people can respond to these therapies for prolonged periods of time. And this is just an example of where it works the best in metastatic melanoma, where people have had disease-free intervals measuring in years. But getting back to the whole question about clinical trials, whenever I talk to patients about clinical trials, I think there's some important questions to think about. And the first question to talk to your doctor about is there a clinical trial available? What I can tell you is every institution is working really hard to try to get patients access to these experimental agents. And here at the Farber, Peter works really hard to try to get clinical trials for all the gastroesophageal cancer patients. But it's challenging because these trials open and close. So sometimes there are gaps. So it's the first thing to ask your doctor is there a clinical trial for my cancer? And really, the more nuanced question there is there a clinical trial for where I am in my cancer battle? Like perhaps there's a clinical trial if you just got diagnosed. Is there a clinical trial if you've already been treated with one line of therapy? Is there a clinical trial if you've been treated with 3 lines of therapy? Then, if there is a clinical trial, the next question is to ask your oncologist, do you think this is worthwhile? Is this something you'd recommend? I have a lot of people asking, is this something you'd put one of your family members on? I think those are really fair and honest and appropriate questions. The other thing, you always have to think about what the disadvantages are, because as excited as we are about clinical trials, the honest truth is many times these don't work. So you want to know, what are the side effects? What potential harm could happen? What I can tell you is every clinical trial has a consent form usually about this thick, that will outline what the side effects are, but also will talk about how frequently you'll be in the hospital, how frequently you'll be getting these treatments, because another cost of clinical trials is your time. When you're getting treated with standard therapy, your oncologist will do the minimum number of visits to the hospital that you need to get those treatments. So you might come in every 2 weeks to get FOLFOX. Whereas if you were on a clinical trial of FOLFOX, that trial might make you stay, instead of being there for 4 hours, you might have to be there 8 hours for experimental blood tests, that you might have to come once a week. So there's a lot of extra time, a lot of extra visits that go on with a clinical trial, and sometimes even tumor biopsies. So it's important to ask about those questions. Another question, another very important question, is there a placebo? And one thing I want to reassure everyone is, if you go on a clinical trial with a placebo, your doctor has to tell you about that up front. So it won't be that you sign up for a clinical trial and they're keeping it a secret from you. You will know up front if there is a placebo or not a placebo. Another thing about clinical trials is, will your insurance cover the costs? And what I mean by that is, when you go on a clinical trial, we think of two different costs. We think of standard of care costs. So if you were getting treated with chemotherapy, say FOLFOX, your insurance company would pay for the doctor's visits, the blood tests associated with FOLFOX, the FOLFOX drugs, and the CT scans. If you go on a clinical trial with FOLFOX, the way it works is your insurance company would still pay those standard of care costs associated with the FOLFOX, but anything extra, and what I mean by extra is some trials require tumor biopsies, some trials require specialized blood tests where we're testing the level of the study drug in your blood. Some trials will give you an experimental drug. All those extra things that aren't standard gets charged to the pharmaceutical company. Getting to the standard cost that you would have gotten anyway, the ordinary blood tests, the ordinary CT scans, the FDA-approved drugs. In Massachusetts, there's a state law that your insurance company has to pay for the standard of care cost. I know there's some people probably from out of state, and I should say, in some states, one state that comes to mind is Kentucky, because I had a tough time with a patient from Kentucky, where this patient from Kentucky wanted to do a trial, but their insurance company was not willing to pay the standard of care costs. So really what I would say for this Massachusetts-based audience is, most of the time it's no problem. Most of the time your insurance company, actually in Massachusetts all the time, your insurance company has to pay for the standard of care costs. But if you're out of state, it's just important to ask these questions. Once you've decided to do the trial, the next step is that there's eligibility screening. And what I mean by eligibility screening is, when a clinical trial is designed, they have to select a population where it's the right thing to test them in that drug. So for example, going back to the TOGA trial, if you didn't have that marker, the HER2, you weren't eligible for that trial because we knew that wasn't going to work anyway. So that's one type of eligibility screening. Another type of eligibility screening is just making sure it's safe. So for example, in the TOGA trial, it caused heart problems. In small numbers of patients, it can cause heart problems. And so what they wanted to do is just want to make sure that if you had a pre-existing heart condition, that you didn't go on that trial because it could have put you at too much risk. And I just wanted to briefly end by just saying the types of clinical trials that there are. And one of the ways I think about these clinical trials is that there's some trials where you'll get FOLFOX. So if Dr. Enziger said when you ordinarily get gastroesophageal cancer, the first therapy you get is FOLFOX. And the trial will make it so that you either get FOLFOX, which you would have gotten anyway, so it's really not a hard sell, or you'll get FOLFOX in the experimental agent. So one trial that Peter's running right now is a really novel therapy where it's attacking a protein called FGFR2. And there's been some very promising results. And if this trial works, this therapy will actually become FDA approved. It's a phase 3 trial. And the way it works is, when someone's newly diagnosed, that's a key detail, when someone's newly diagnosed with gastroesophageal, metastatic, so it spreads to different parts of the body, when someone's newly diagnosed with metastatic gastroesophageal cancer, they can enroll on this trial. They can even start the trial, but they only can continue on the trial if they have the right protein. And again, the reason for that is we know it's not going to work if you don't have the right protein. So what Dr. Endiger would do if he was treating you is he would screen your tumor to see if you had that protein, and if you did, you could continue on the trial, and if you went on the trial, you would have a 50% chance of just getting regular FOLFOX, which would be fine because you would have gotten that anyway, or getting FOLFOX and the FGFR inhibitor. Other types of trials are randomizing either to get an experimental agent or placebo. So one trial we did here was for patients who had esophageal cancer resected, and ordinarily after someone has an esophageal cancer resected, we don't give additional therapy. We wanted to see if immunotherapy would work in this space, and the trial was comparing patients who got standard of care, which happens to be no therapy, so placebo, or getting the immunotherapy. Other types of trials include trials where you're just automatically getting the experimental agent. You're getting, in this case, a PARP inhibitor and ramucirumab, or in phase 1 trials where you'll definitely get that type of study. One very important point I wanted to just drive home is that clinical trial portfolios are very dynamic. What I mean by that, if you talk to your doctor and they tell you, we don't have a clinical trial for you, I think it's important just to keep asking because we're always opening trials all the time. So if the answer is no today, it might be yes in the same circumstance in 4 weeks just because these trials are always opening and closing. And just to sort of finish up, I was just going to run through just 3 vignettes about different patients in their cancer journey and what trials are open for them here at the Farber. So for a patient with newly diagnosed gastroesophageal junctional cancer, one option is not to do a clinical trial, just to do standard FOLFOX. But the other option is to be screened for 2 markers. One marker, as we talked about, is called FGFR2. Another marker is called Claudin 18. And so the way Peter set up the gastroesophageal program right now is for any gastroesophageal cancer patient with metastatic disease that's spread all over the body, he's sort of set up that when these patients come in, we screen them for both trials because we don't know which marker they have ahead of time. And if they have one of these markers, so say they have Claudin 18, we'll put them on the FOLFOX Claudin 18 antibody trial. Or if they have the FGFR2 marker, we'll put them on the FOLFOX FPA 144 trial. For second-line patients, and what I mean by second-line is those are patients who've already been treated with a first-line therapy like FOLFOX. The standard option is from the Rainbow trial is a regimen called Taxol and ramucirumab. However, what we've been doing is especially for patients who have something called DNA repair deficits, and we take this from breast and ovarian cancer. So in Dr. Yergelin's talk, he might have talked about that some patients have difficulty repairing DNA, and because of that get gastroesophageal cancer, things like BRCA1 and BRCA2 mutations. And if they have these DNA repair deficits, we've been putting them on a clinical trial of a drug that's FDA approved in ovarian cancer for patients with those mutations, and we combine it with ramucirumab. And finally, for patients who've gone through our best therapies, who've gone through FOLFOX, Taxol, ramucirumab, and pembrolizumab, for those patients, what we're doing is we're putting them on phase 1 trials, and to select the phase 1 trial, we're using results from the molecular profile. And I wanted to conclude there, but I just want to thank everyone for listening.
What's the rate of conversion from phase 3, it's a hard question to answer, phase 3 to FDA approval?
Right, so I can repeat that. So the very good question is, what's the rate of approval from phase 3 to FDA approval? The rate there is about 15 to 20%. What really, I think, drives home how hard this is the rate from phase 1 to FDA approval is somewhere between 3 and 5%, so it's low. Good question.
Any other questions for James? Please remember you can always write down your questions and we can ask them during the panel. Okay.
Can we have access to any of the slides?
Yes, so these slides and the talks will be put on 2 websites. One will be the Dana-Farber website, the Esophagogastric Cancer Center website, and then also Debbie's Dream will have it on their website. So you can listen to James every night if you like.